There we go. So, welcome to the lecture for cellular respiration and photosynthesis. So, this chapter is going to look at those processes in a bit more detail than we've done before. Let's start off with this picture right here. Who is this? This is Olympian Michael Phelps. I believe he has the most gold medals in Olympic history. For swimming, that's what he has his medals in, and when he trains, he burns quite a few calories a day. So we're not going to look at the technical definition of a calorie in this class. We're simply going to know the fact that calories are effectively a measure of chemical energy in the food that we consume. Technically, calories are a lot smaller than what we actually measure. We measure what are called kilocalories, which consist of 1,000 calories. So every time you eat 1 calorie, you're actually eating 1,000 technical calories. But Michael Phelps, he trains for the Olympics, and when he does, he burns a lot of calories swimming, which means that he has to consume enough calories so that he doesn't lose that muscle mass that gives him the strength to swim so fast. The average person is supposed to consume between 2,000 and 2,500 calories per day. That is the general dietary recommendation, but it really depends on your activity level. When I was in grad school, I worked at Walmart and was on my feet for 8 hours a day, and when I didn't work, I usually played basketball about 2 or 3 hours every night. I consumed between 3,000 to 4,000 calories a day, and I still lost weight during that time. Nowadays, I'm not on my feet nearly as much. My diet has been reduced quite a bit, and I'm staying at a pretty steady weight even though I eat way less than I did back in grad school. So calories in, calories out. Michael Phelps, when he trains, consumes somewhere between 10 and 12,000 calories a day. And if you ever get bored, go online and look up the foods that he actually ate. Massive, massive meals. And he didn't gain any weight from it because every day when he trained, he burned most all of that off in the process. So, obviously Michael Phelps used a lot of energy to swim. His cells needed the energy to perform that work. He got it from his food, and so he had to consume quite a bit to get the energy. The process by which Michael Phelps's body broke down the food energy and made it available for his cells to use, that's cellular respiration. That's a method of harvesting energy. The other method is photosynthesis, which we will also look at here. So some of this, hopefully, you remember from the previous chapter. Cellular work requires energy. Food contains chemical energy, a type of potential energy stored in the bonds of the atoms in food. This is the energy that we must get. We are heterotrophs, right? We do not make our own food, and so we have to consume food, like the cantaloupe and peanut butter sandwich in this picture, in order to get energy. If you remember though, food energy is not directly usable. You cannot directly tap into food and use that energy to power your cells. Your cells have a very specific currency they must use in terms of energy, and that's ATP. And so cellular respiration, which we're going to look at here first, involves the breakdown of food, which releases that stored energy, that stored chemical energy, and converts it into ATP, just like a currency conversion you would do. That ATP is then usable by the cells, and cells directly tap into that in order to have energy to do work. Again, we defined calories, and we talked about Michael Phelps's calorie intake. So looking at cellular respiration, here's a formal definition for you. Cellular respiration is a metabolic process in which energy-rich food molecules are broken down and their energy is stored in the form of adenosine triphosphate. So cellular respiration itself is not powering your cells. Cellular respiration is breaking down food to release the energy and convert it into a form that can be used to power your cells. Here is the formula for cellular respiration. You'll definitely want to be aware of this, all right? So, what do we have here? We have, let's see if I have it written. Nope. Over here we have glucose and molecular oxygen. You eat food, all right? A lot of food has sugar or glucose present in it. This is also the chemical formula for for fructose. So other sugars can be used here as well. Glucose plus oxygen, you have to breathe oxygen for this reason, is converted into carbon dioxide and water. Glucose and oxygen, these are what we start with. They're called the reactants. The reactants are what go into a chemical reaction. The chemical reaction occurs, the bonds are broken, and new bonds are formed, and we end up with products that are very different from the initial reactants. First law of thermodynamics tells us that energy is not created nor destroyed, which means that the energy that was in glucose wasn't destroyed in this process, it was converted. It was lost out of this reaction and moved to ATP. Another term for you is the word aerobic. You hear this word in everyday usage as a type of exercise, but it's kind of appropriate there because if you have aerobic exercise, I believe that's another term for cardio. And so you're using your lungs, right? Well, in this case, aerobic simply means it requires oxygen. So if oxygen is needed for something, it is said to be aerobic. So cellular respiration is an aerobic process. The alternative to being aerobic is to be anaerobic. So some living things are anaerobes. They legitimately cannot survive in the presence of oxygen. And so you'll find them in certain what are called anoxic environments, oxygen-free environments on Earth. But aerobic means oxygen involved, anaerobic, no oxygen. If you think back to the previous chapter, we talked about endergonic and exergonic reactions. An exergonic reaction is one where the product has less energy than what we started with in the reactants, which means some of that energy got released and went to somewhere else. In the case of cellular respiration, the energy that is released from the sugar is stored in ATP once it gets released. So, cellular respiration means that the reactant has a high amount of potential energy while the end product has very little. Energy is not created or destroyed. The energy that was in sugar is put into ATP. Almost all organisms conduct cellular respiration, and it's important to remember that that includes the organisms that produce their own food. Animals like burrowing owls, plants like this aster here, fungi as well, even unicellular creatures like protists and bacteria. One important thing to note is that plants conduct cellular respiration even though they make their own food, right? Because if you're a plant, you can make your own food, but you still have to convert that food into usable energy, which is ATP. So plants will do photosynthesis, and then they'll engage in cellular respiration once they've made that food. So, mitochondria. The mitochondria is actually a plural term. The singular version for mitochondria is mitochondrion. The mitochondrion is an organelle found in your cells. Right? It looks something like this. Let's go over a little tour of a mitochondrion. Powerhouse of the cell, right? It is in a way. It's where this whole thing happens. It's converting food into ATP. So let's take a look at the structure. There's obviously the outer membrane and an inner membrane. The part that's bright yellow is the intermembrane space. So we see how there's the outer membrane and then the inner membrane, which has these folds, and then the space between them is called the intermembrane space. That's the bright yellow. The mitochondrial matrix is the one that's kind of a tannish color. Here on the inside, I believe it is filled with a semi-fluid medium. And then there's these folds. These folds in the inner membrane. They are called cristae. What is their purpose? Why is the inner membrane not just the same general shape as the outer membrane? Because having folds increases available surface area for the inner membrane. All right? The inner membrane, if you were to stretch it and the outer membrane out and put them next to each other, the inner membrane is going to be longer. Right? Because those folds increase the surface area. And that's important because something obviously quite important is occurring at that inner membrane, and we'll get to that. So that is the general structure of the mitochondria. So you consume sugar, and for every sugar or glucose molecule that goes into cellular respiration, one glucose goes in. What comes out is 36 to 38 molecules of adenosine triphosphate, or ATP. So one glucose is put into this, 36 to 38 ATP come out of it. You can also use molecules other than glucose. For instance, if you eat an apple, that's primarily fructose, that can be used as well. Even proteins can be used to ultimately produce ATP as well. Things other than sugars. It's a 4-step process: glycolysis, the transition reaction, the Krebs cycle, and oxidative phosphorylation. So we're going to look at what happens during each of these 4 steps. Starting with glycolysis. Glycolysis is stage 1. This is the only stage of cellular respiration that occurs outside of the mitochondria. So this is occurring in the semi-fluid interior of the cell, known as the cytosol or the cytoplasm. The word glycolysis means splitting of sugars, and that's a pretty apt name when you think about it. You're splitting up a glucose molecule, and by the end of this, you're going to have 2 molecules that only have 3 carbons each instead of 6. There are 2 phases to glycolysis: energy investment phase and the energy payoff phase. I want you to think of this in terms of money. If you're going to invest in something, you have to put some money into it at first, so you're going to go kind of in the hole. You're going to be losing money initially, but the hope is that you'll gain something on that investment to where you'll end up making more money than you initially put in and have a profit. Same thing with the energy investment and payoff phases for glycolysis. The investment phase involves spending some ATP. Remember, ATP equals the energy currency of the cell. And so when you spend ATP, actually I'm going to draw it out. If you guys remember, ATP, it's triphosphate. Most of the energy is in that last phosphate bond. When you spend ATP, what you're doing is you are breaking apart that last phosphate bond, which releases energy that is used for cellular work. And so if we have ATP and we break off that phosphate bond, we're basically spending the energy that was stored in it. Think of it like this might be a poor analogy, think of it like a battery. This is a charged battery. This is a battery that's dead, but it's rechargeable. Instead of using electricity to charge it, though, we're going to use food. So that's kind of the general idea. So energy investment phase, we start off by spending 2 ATP. The energy payoff phase, we create ATP, and I assume we're going to create more than 2 because we need to get a good payoff for that initial investment. So let's take a look at what happens during the energy investment phase. During this phase, we start off with glucose. So I'm going to illustrate up here a very simplified, and this is the same way that your textbook would illustrate this. We're not showing any of the hydrogens or the oxygens. All we're showing is the carbons. And if you remember, glucose, C6H12O6. So C6, 6 carbons. This is what we start with. So during the energy investment phase, 2 molecules of ATP are spent. Therefore, we need we need some energy to begin with. Just like if you want to invest in something, you have to have some money up front. So in order to conduct glycolysis, the cell must have at least some energy already available in the form of ATP. So we've got our glucose. ATP comes along and what it does is it releases additional energy to our glucose molecule. Glucose already has a lot of energy. Glucose already has a lot of energy here. We've just given it more, right? And we've given it so much energy that it's actually unstable at this point. So ATP jettisons one of its phosphate groups, which latches onto our sugar molecule here. Another molecule of ATP does the same thing. This original ATP had a lot of energy. Once it becomes ADP, it doesn't have that energy anymore. It dropped off the energy onto our sugar molecule. And by the time it's done that, we've made the sugar unstable. Here's an analogy to help you think about it, hopefully. If you've got a stack of books that's this tall, it's not quite unstable necessarily at this point, but if you add more books to make it this tall, now it's really quite unstable. It was already a tall stack of books. By adding more books, you've made it even less stable. How can you make it so that that stack of books is stable? Well, one thing you can do is you can split it into 2 separate stacks, each of them only this high. You can split the stack of books. And so because ATP has released energy to our glucose, which was already high energy to begin with, it has now supercharged it to a point where it's not stable as a 6-carbon molecule. So what it does is it splits. It splits into these 3 carbon molecules, a 3 carbon molecule intermediate. Now, by the time it gets even to this stage right here, it's no longer glucose anymore. All right, it's a different form of molecule because of the fact that this chemical reaction is occurring and changing some of the bonds. I'm not concerned that you know the names of all of the intermediates between glucose and our end product, but I do want you to be aware of the definition of the word intermediate. It's a molecule that occurs in a chemical reaction between the initial reactant and the final product. So these are intermediates in the process of glycolysis. Starting reactant, intermediates. By the time we've got these 3 carbon molecules, we are done with the energy investment phase. All of this is the energy investment phase. The name of this particular intermediate, each of these, are called G3P, glyceraldehyde 3-phosphate. So 2 molecules of G3P exist at the end of the energy investment phase. We're currently standing at -2 ATP. We've spent 2 of them. We haven't produced any new energy currency yet. The energy payoff phase, the second half of this, is going to give us 4 molecules of ATP. Therefore, if we're at -2 right now, and we gain 4, we'll have a net gain of 2. 2 ATP are made by the end of the energy payoff phase. A couple of other things are going to happen as well. Byproducts. A byproduct is something that's going to be made in addition to the final product, and there are several of them in glycolysis. For instance, there are 2 water molecules produced as a byproduct. And then there's something called NAD+. This is kind of a tricky thing. NAD+ stands for nicotinamide adenine dinucleotide. You do not have to know that. But here's what you do want to know. NAD+ is an electron carrier. What does that mean? What it means is NAD+ goes up to a molecule, plucks an electron away from that molecule, and carries it off somewhere else. Electrons in our G3P, that's what we have at the start of the energy payoff phase here. Electrons in these 2 molecules have a lot of stored energy. There's a lot of energy available there. So what NAD+ does is it comes over to our G3P intermediate, and it's going to pluck a couple of electrons away from these molecules. when NAD plus gets the electrons from our intermediate, it becomes NADH. And when it's NADH, that means that it has electrons with it that have a lot of energy. It's like it's become charged with energy from these electrons, although it's just carrying them. But it plucks some electrons away from our intermediate here and carries them off somewhere else to be a source of energy at a further future time. So, NAD plus, electron carrier, comes in and removes electrons from our sugar intermediate, becoming NADH. It's not going to actually do anything else at this stage, but it has gone off and carried those electrons somewhere else, full of energy. 2 NADH are going to be made during this glycolysis phase. The electrons in glucose are a source of energy. By carrying the electrons, NADH is effectively relocating bundles of available energy to another location. So after the energy investment phase, where we lost 2 ATP, we had the energy payoff phase, which was a series of chemical reactions. And by the time we get to the end of the energy payoff phase, we are left with 2 molecules that have 3 carbons each. And these molecules are called pyruvate. So we have effectively split the original glucose into 2 molecules of pyruvate. And those 2 pyruvate will enter the second stage. Let me make sure that, my phone is still recording. I'm going to stop the recording here because it's at 27 minutes. And we'll start up with the next stage in the next video. All right, welcome back. So, we ended at the end of glycolysis. We ended with 2 molecules of pyruvate. Let's just make a quick list of what the byproducts were for glycolysis. The byproducts of glycolysis were 2 ATP. That was our net gain. 2 water molecules, 2 NADH electron carriers. The water your body might hold on to or get rid of depending on your hydration status. ATP, those are ready to be used as currency for the cell to power the cell's work. The NADH are carrying electrons full of energy off to some future endeavor. Which brings us to the transition reaction. Another name for this one is pyruvate oxidation. So pyruvate was the name of the molecule that was at the end of glycolysis. So by the time glycolysis was done, we had 2 molecules of pyruvate. These were produced outside the mitochondrion in the cytoplasm. Now they enter the mitochondrion once we have them at this point. So 2 pyruvate are going to enter the mitochondrion. This is much better shown as a picture, which unfortunately I do not have up here. So that's okay. We'll draw it. What happens during this transition reaction is that each molecule of pyruvate is going to be oxidized, which means it loses electrons, to 2 molecules of what are called acetyl-CoA. So by the time we get to the end, this whole thing is going to be the transition reaction. By the time we get to the end product, we will have to have done a number of different steps. Notice the first major thing. We've gone from 3 carbons in pyruvate at the beginning to 2 carbons in the acetyl coenzyme A, which is what CoA stands for, at the end. That tells me that somewhere along the line here, we have to lose a carbon. And we do. We lose it in the form of CO2. You're breathing it out right now, and part of what you're breathing out comes from your cells conducting this transition reaction or pyruvate oxidation. So CO2 leaves. Another thing happens as well during this. NAD+ is once again going to come in and snatch some electrons up. So NAD+ comes in and takes some electrons from each of the pyruvate molecules. Thus, it's taking that source of energy, those electrons, off to some future thing. So during this step, 2 pyruvate are converted to acetyl-CoA, and in the process, 2 byproducts are made. 2 NAD+ pick up electrons full of energy to carry them off somewhere else, and 2 carbon dioxide are going to be produced, which ultimately will just leave the body as a waste product. So by the time this is done, we have 2 molecules of acetyl-CoA. Which leads us to the third stage, the Krebs cycle. It's also called the citric acid cycle. This occurs in the center matrix of the mitochondria, that fluid central matrix. I could try and explain it, but I think a picture is worth 1,000 words for this particular stage. All right, let's go through this. First of all, it's a cycle, which means something will always be coming back around. And that something is called oxaloacetate. Oxaloacetate will always be regenerated in this cycle, and it will come back around as the new molecule enters. The 2 carbons of acetyl-CoA enter this Krebs cycle, and the oxaloacetate coming back around is joined with them. 2 carbons come in the cycle. Oxaloacetate is a 4-carbon molecule. They join together to form the 6-carbon molecule known as citrate. The citrate undergoes a series of chemical reactions, ultimately being transformed back into oxaloacetate, and the whole cycle continues as new acetyl molecules enter the cycle on a continuous basis. As far as what you need to know from this, you're going to want to know what are the byproducts. Let's take a look. First of all, keep in mind, we've got 2 molecules of acetyl-CoA. This image is showing you what happens to one of them. So everything that gets produced as a byproduct here, we have to double the number that we have, because this is going to occur twice. So byproducts made in the Krebs cycle. Let's start with CO2. I noticed that there are 2 molecules of CO2 that ultimately jettisoned from the cycle. If this is going to occur twice, that must mean that there's going to be not 2 CO2s, but 4. So most of the carbon dioxide that you are breathing out comes from your body's Krebs cycle. 4 CO2. Here's some more NAD pluses coming in, removing electrons, and carrying that source of energy off somewhere else. I see 3 NAD pluses coming in and removing electrons, which tells me this is happening twice. There must be 6 of them. Also, there's something else here, similar to NAD+, but not quite. FAD, flavin adenine dinucleotide. Different molecule, same exact function as NAD+. It comes in, gathers some electrons, carries those energy-rich electrons off somewhere else. Since there's one FAD molecule which takes up electrons and becomes FADH2 when it does that, there must be 2 in total because this is happening twice. And last but not least, ATP. So, 1 ATP is produced per churning of the cycle. Since the cycle churns twice, 2 ATP are made. So, thus far, we're through 3 of the 4 stages of cellular respiration. We have made a grand total of 4 ATP molecules, 4 pieces of energy currency. We've also created some waste CO2 and created a whole bunch of these mobile electron carriers moving around with energy-rich electrons in their grasp. I'm going to guess that the last stage must utilize them somehow, because that's where a whole bunch of this energy that came from your food in the first place, that's where it now is. It's held by these mobile electron carriers. So what happens in the next phase? Well, let's get into it. This next one, and this just talks about what I've given to you on the board. Tells you all about what happens. The last phase then is going to be oxidative phosphorylation. Oxidative phosphorylation. This is the primary workhorse in producing ATP. 32 to 34 ATP molecules are going to be made here. This is easier shown than written. Please listen up. This is a tricky thing for a lot of people to get. So the first thing that's happening is right here. NADH and FADH2, all of those that were produced during the first 3 stages are going to come here. Okay? And what they're going to do is they're going to release their electrons. And actually, before we get into that, I want you to have a visual of where we're looking at in the mitochondrion. All of this is happening inside of the mitochondrion. There's our outer membrane. Here's the folds of the inner membrane. This picture right here is showing us a snapshot of roughly right here. Right? What it's showing us is all this in here is the mitochondrial matrix. I know this is going to look really small on my phone camera, but the mitochondrial matrix is this part right here. We have the inner membrane, which is this right here, and then above that we have the intermembrane space. That's this right here. So we're looking at that part of a mitochondrion. So keep that in mind. So, NADH and FADH2 release their electrons. This picture is my hypothetical energy staircase. If you're at the top of it, you have a lot of energy. If you're at the bottom, you do not have much at all. The electrons released by NADH and FADH2 have a lot of energy. They are at the top of this staircase. Energy cannot be created or destroyed, but the energy can be released. So, on the staircase, we've got a series of molecules which are called electron carrier proteins. What happens is the electron is passed from one protein to the next, and every time it gets passed, it loses some energy. I want you to think about the game of hot potato, like a literal game of hot potato. First person who gets the potato, it's the hottest for them. They pass it to someone else. They pass it to someone else. By the time it gets to the fourth person, it's not as hot anymore. The heat has dissipated. Every time it's been passed from person to person, some of that heat is lost from the potato itself. Every time the electron is passed from one carrier to the next, it loses energy and effectively goes down the energy staircase. And by the time it gets to the very end, where there's an oxygen waiting for it, it doesn't have much energy left at all. By the way, the only reason that this molecule even goes down the staircase is because oxygen is pulling on it. Oxygen, the oxygen you breathe, the whole reason you need to breathe is because you need that oxygen to be there to pull the electron down this staircase. That's why you need oxygen. So, the electron, as it goes down the staircase, loses energy. The energy is released by the electron. Energy is not destroyed. It is merely transformed. And so what is the energy released by the electron used to do? It's now available. It can be used to perform some type of work. And what it's used to do is move protons over to one side of the inner membrane. In particular, it's used to move protons against the concentration gradient so that the intermembrane space is full of them. This can be a little tricky concept to understand, I know that. But energy released by the electrons that are electron carriers dropped off, energy released by that electron is used to do active transport to move protons to one side of the inner membrane. Basically, you are loading the intermembrane space here full of protons. The more protons you put in that space, the greater the potential energy that you have becoming stored in the intermembrane space. Think of it like this. The more protons you pump into the intermembrane space, it's equivalent to making a dam higher and higher and higher and higher. The higher you make the dam, the more potential energy you're going to have in the water held behind it. Likewise, the more protons you pump into the intermembrane space, the more potential energy we've now got stored in there because of the discrepancy in how many protons are here versus the other side of the membrane. Once you've got all of the protons, or a huge number of them at least, in the intermembrane space, then we are ready to actually make ATP. Just like you can take water flowing over a dam and convert it to electricity through hydroelectric power, so can you take the movement of protons back into the matrix and use that to produce ATP. It's a tricky concept, I know, for a lot of people to understand, but this is how your body converts, ultimately, food energy into usable energy in the form of ATP. The actual enzyme that produces ATP using the protons moving across the membrane, it's called ATP synthase. And the name of that process that it does is called chemiosmosis. I'm not worried that you have a specific definition of this, but if I were to explain this process, you'd want to know that chemiosmosis is what I'm referring to. The first part of this whole process, the passing of the electron along this chain of carriers, that's called the electron transport chain. Often that term is used to refer to the whole shebang as well, but be aware of that term, you'll run into it. This is done to such a degree that we can make 32 to 34 ATP using ATP synthase and using the protons and their movement across the membrane as our energy source. This is the bulk of where you make most of your ATP from. The next slide just explains it all. You're more than welcome to read it. I'm not going to read through it again. The electron transport chain is said to be, or cellular respiration in general, is said to be aerobic. If something is aerobic, it requires oxygen. And so oxygen is necessary specifically to pull on the electrons in the electron transport chain. There are a number of toxins that are actually enzyme inhibitors. I don't know if you guys remember, hopefully you do, what an inhibitor is. It's something that blocks an enzyme's function. So, a couple things that you do not want to breathe in are actually toxic because they inhibit one of these carriers along the electron transport chain. Carbon monoxide, for instance, inhibits one of these carriers, which prevents the electron from going down the chain. It prevents the energy from being released. It prevents the protons from moving appropriately. You don't make ATP. It kills you after a very short amount of time. Cyanide is another toxin that works as an inhibitor in this process. So these kill you by inhibiting cellular respiration, and specifically inhibiting the last part of it. Let me see how much time I got on my phone right now. Nope, I still got time. Fermentation. So if there's no oxygen, you can't conduct cellular respiration. However, there is a way to make ATP from food even if there is no oxygen available. That is fermentation. So if there's no oxygen, it is anaerobic. Fermentation is a method of producing ATP from food without the presence of oxygen. That sounds like an upgrade to me, right? If you can do this whole thing without needing oxygen, why would you ever do cellular respiration? Why? Well, the reason is because in cellular respiration, one glucose yields roughly 36 ATP, and that's a low estimate. In fermentation, one molecule of glucose is going to yield 2 ATP. Now, I don't know about you, but if I invest $1 in the bank and I can get $36 back, I like the sound of that a lot better than investing $1 and getting $2 back. However, you know what $2 is better than? Nothing. So, fermentation does offer the benefit of working under anaerobic conditions, but the yield is not nearly as efficient as cellular respiration is. There are some bacteria and some other organisms that might live deep under leaf litter or in deep oceanic environments where there isn't enough oxygen or isn't any oxygen. And these organisms have evolved to the point where they are not able to even live in the presence of oxygen. It's fatal to them. They are called obligate anaerobes. The only thing they can do to utilize energy is to ferment. They are incapable of conducting cellular respiration. However, a number of species, including yeasts, are what's called facultative anaerobes. These guys are the best of both worlds in terms of energy harvesting. They can do both. If there's oxygen, cellular respiration, it's much more efficient. If there's no oxygen, we'll switch to fermentation. So yeast is one example of a species that does this. In the lab that I hoped we were going to do, we were going to actually conduct fermentation using yeast and sugar compounds and measure the rates of fermentation. But unfortunately, we never got a chance to do that. That was going to be your 4th lab report. So I might have you write a 1-page summary over something fermentation related in place of that, but stay tuned. Stay tuned for that. What does fermentation do? It actually uses the same exact chemical process as glycolysis. If you remember glycolysis, it did not happen in the mitochondrion, and it didn't require oxygen. Only the electron transport chain needed oxygen. So fermentation uses glycolysis, and then that's the end of it. Remember, glycolysis had a net gain of 2 ATP? Thus, so does fermentation. Fermentation yields us 2 ATP per molecule of glucose. Now, in order for fermentation to work, at the end of it, NADH has to release the electrons and be regenerated as NAD+. If you remember, the whole point of NAD+ becoming NADH, being reduced to NADH, was that it had energy in those electrons that it would use at the electron transport chain at the very end of cellular respiration. Since fermentation just ends after glycolysis, NADH doesn't have to do that same purpose anymore, and so it has to jettison electrons, become regenerated as NAD+, and there's a couple different ways that fermenting organisms do this. The 2 different methods are called lactic acid and alcohol fermentation. Let's take a look at those 2. Lactic acid fermentation. In lactic acid fermentation, the 2 pyruvate molecules are reduced to 2 lactate molecules. Because remember, in glycolysis, we start with 1 glucose, and we end with 2 pyruvate molecules. And that's how it is with lactic acid fermentation. 1 glucose to 2 pyruvate, but then at the end of it, those pyruvate are reduced to lactate molecules. Certain foods use lactic acid fermentation in their production. Yogurt involves lactic acid fermentation with milk, dairy products. Sauerkraut involves the lactic acid fermentation of cabbage. And to me, it's just a disgusting thing. I've never, don't understand the appeal of sauerkraut. But keep in mind with fermentation, we use it to produce foods. But in truth, we're just using the byproducts to our benefit. It's actually a process that organisms are doing to produce usable energy, to convert energy into a usable form. Yogurt, sauerkraut, we are consuming food products that ultimately have been modified by the byproduct of bacteria converting food into their usable energy form. It's kind of interesting thought. Alcohol fermentation is a process that also uses glycolysis as the method in which the ATP are made. By the end of it, the pyruvate are converted to 2 molecules of ethanol, and carbon dioxide is released as a byproduct in this process. So this is done usually by yeasts or bacteria, and if you've ever used yeast in baking bread, the yeast conduct fermentation during that baking process, and the carbon dioxide bubbles that they produce as a byproduct is what causes the bread to rise. That's what causes the air bubbles in the bread. Also, of course, alcoholic beverages are produced through alcohol fermentation. Different sugar sources for the yeasts that are used ultimately results in different drinks. For instance, if you take grapes and give and allow organisms to ferment the grapes, that'll produce wine. If you give barley for the food source, it would produce beer. Potatoes produce vodka. I want to say molasses produces rum. And so that's really where you get all the different drinks from. It's from different sugars being given to organisms to ferment. And the ethanol in those drinks ultimately is what causes people to act a little strange when they consume them. It's what affects our behavior when consuming alcohols. So alcohols and bread with yeast, those would be examples of fermentation. Yeast and bacteria are the 2 principal species that actually conduct fermentation that is used to produce our foods or drinks. All right, I think I'm going to stop this lecture here and start a new one for photosynthesis.